Today we're going to be talking about the measurement of the brain, and we continue. And you'll see it is very simple yet very difficult. Did you do the readings? The readings from your textbook. And of course you'll see the pictures in the textbook as well. Last time we talked about the billions and trillions of neurons and the glial cells and the movement of ions and the electrophysiological changes. And we talked about how we can measure each one of them. This is not easy. It is a very complex process. But we also gave you the good news that the brain is similar with the animals on the lower phylogenetic scale. The activities are similar. The neurochemicals are similar. The ionic movements are similar. The neuroanatomical sites are similar. So that is good news. And of course, the good news is also that technology has advanced so that you have more and more available measures of the brain. Whether you're talking about measuring ions or whether you're talking about the movement of molecules or the electrophysiological changes. So the good news is that we have similar organizational structure, similar chemical composition, similar organic and ionic dispositions, and at the same time these are grouped together. And of course, brain cells are similar to other cells in terms of nutrients, and of course we can study them in vitro and in vivo. In vitro is outside in a petri dish, or outside the body, living organism, or in vivo, which means alive within the system. We have developed different techniques, and of course there is a consistency. Nature is extremely consistent, you must realize that. And the consistency of nature, the consistency of natural organisms include even the behavior of cells. So that consistency works to our benefit. Whether it is ions, whether it is neurons, whether it is ganglionic cells, whatever it is, they work in the same manner consistently and respond in the same manner. Now this is the good news which ends here. The bad news is, of course you understand, the bad news is the brain is extremely complex. This little scalp has extremely extraordinary cells which are firing away, and they are intricate networks. So, what do? We identify, it is difficult to identify the networks. It is difficult to identify that if there is one process involved. It is impossible that there is one process involved. There are several connected processes which work together, and there are several levels of functioning: chemical, physical, physiological, electrical, ionic. How do we know where one is interacting with the other? It is difficult to measure simultaneously. Then, brain functions both at the molecular level and at the molar level. At the molecular level, you have the neurons firing away, Assume this is a neuron, this is another neuron, and they have these connections. They're firing away. And there are little molecules running between these neurons and these synapses. But translated, it is this behavior that you see. My talking, my movement of hands, my attending to you, and you attending to me. This is the movement of molecules translated into gross actions and behaviors. Now we have to measure this at all these levels, the molecular and the molar level. Measurements of these behaviors can be made at each of these levels. And these tiny, small molecules and their communication with each other, and the movement of little ions running back and forth, and the amino acids which are changing, we can measure that. And of course, measuring that does take some techniques. Measuring also translated into behavioral changes. You must understand how to link the behavioral changes with these ionic changes. It is not easy because you have to know beforehand, after you've done your studies, whether these ionic changes correlate with the behavior that you are trying to study. And then there are many interconnections and many neurochemicals working in one location, in one setting, and working simultaneously. So you have to measure each one of them to really make a statement that yes, this is the neurochemical which is responsible for this behavior. Neurochemical dopamine responsible for schizophrenia. Neurochemical serotonin responsible for hallucinogens, and so on and so forth. And isolating one does not give you a complete picture. So you have to have a complete picture. And let me tell you, you can do that. Isolated study gives you only one part of the jigsaw puzzle. It does not give you the complete picture. Let's look at the techniques that are there. After giving you all these good news and the bad news, let me tell you that there are many techniques which are still doing a very good job at identifying brain behavior relationships. Techniques vary in several ways. One, what are we working with in terms of behavior? Are we working with simpler behaviors or are we working with more complex behaviors? Behaviors related to brain areas at the lower level, at the level of the spinal cord, medulla, hypothalamus, or at the higher order cortex, which is very complex. And it's not easy to identify exactly how these areas work together. So, you're working, for example, with motor movement. If I walk, remember I used to tell you in your earlier classes that the basal ganglia at the lower area and the cerebellum are the two areas involved in bipedalism, in walking. So you balance when you walk. You don't fall over every time you walk. But at the same time, to walk to a targeted area. And the walk becomes purposeful only when the cortex tells basal ganglia or the cerebellum to do so. So, there needs to be a understanding and a study of this at different levels. So, what we are saying is, if you study simpler behaviors, you need different techniques. You study complex behaviors and higher order behaviors, you need different studies and different techniques. Techniques become available, secondly, as more advanced scientific knowledge moves ahead. More techniques become available with the advancement of science and technology. We did not have MRIs and the PET scans and the CAT scans and the functional MRIs before. Now we do. It makes it, in fact, it gives a holistic picture. So we do tests of MRIs along with the neuropsych and the laboratory tests. Then thirdly, what is the goal of your study? That also determines the kind of techniques that are going to be with you or that you are going to be using. Is the goal exploration? Is the goal understanding? Is the goal manipulation? So exploration, as you can understand, is just looking at the brain as things happen, describing as is, exploring the brain. Because understanding is a little higher, where you see functions and then you try to understand why these functions occur. And thirdly, you have the manipulation, the most complex, where you are moving into the brain and making changes so that you see how these changes affect behaviors. And why do we do that? We do this to develop theoretical and analytical frameworks and also to develop an understanding of pathology, if it occurs, or normal development of brain, how it occurs. So this, keeping these in view, these are our goals. Basically, to put it in some words, it is to understand, to control, to simplify, to objectify behavior at the microscopic and the macroscopic cross levels. Let me say it again. Basically, we do all this to understand, control, simplify, and objectify behavior at the microscopic and macroscopic levels. Now, why? This is the reason why you are studying behavioral neuroscience. You are objectifying behavior at the microscopic and the microscopic level. Now, what is the simplest thing that we can do? The simplest possible thing that we can do is measure the neuron, its size, its projections, its interconnections, and its functions, its chemical changes. That's the simplest thing that we can do. So you do structural histological procedures to understand the structure of a neuron. This is the neuronal body, the axon, and the dendrites. Use your hands. So you understand this is the cell soma. And so neuron can be understood to see where these dendrites are proceeding to and branching out and connecting with other neurons. So structural and histological procedures to understand the neurons, their format, their size, their interconnections, their projections, and their functioning, basically. Then the second thing that we can do is measure electrical activity of these neurons. Of course, you will find out later on during the course that neurons are highly charged with electrical ions, negatively charged and positively charged ions, which are moving constantly. So we measure that little electrical activity in the neuron as electrical activity of the brain. Then metabolic processes that are also used as measures. Glucose use. Did you know that every time your neurons fire, they use glucose? Of course you know. You eat food and that food is translated into glucose. Then, of course, you also know that oxygen and blood flow is what is needed to keep the brain and any cell alive. And of course, there are procedures such as radiolabeling, which means radioactivity is injected to see how the glucose and the oxygen and the blood flow is moving within the brain setting. Then there are neuropsychological assessments. Then there are behavioral paradigms. I will go into those details. And of course, the use of technology such as the MRIs, the PET scans and the CAT scans and functional MRIs, etc., etc., etc. So, we'll go over these techniques one by one. If you classify techniques, there are three major categories that you can classify them in. One, which are invasive, two, which are non-invasive, and three, which are post-mortem. Invasive are those which enter, which invade the brain. So, we will enter the brain, bring about changes in the brain, whether we measure the electrical activity of the brain, the chemical activity of the brain, or whether we lesion the brain, we cut the brain. You know, there are many little techniques. And we do that by invading the normal brain. And not the human brain, mind you. The invasive techniques are invariably the techniques which are used on animal subjects in the controlled conditions in the laboratory, and I will go into the details of these. The non-invasive techniques. The non-invasive techniques are those techniques in which we do not actually enter the brain or bring about changes of the normal structure of the brain. And we still can measure and we can still assess what is happening. And these are your MRIs, your EEGs, and all these put together form the group that we call the non-invasive techniques. These are also studying the live tissue in vivo without changing the formal structure and the formal working of the brain. The histological postmortem after the death, of course, that is when our histological or analysis and of the structures of the cells takes place. So, these post-mortem studies are done with animals. After we have manipulated the brain in the invasive technique, then we go and verify what we have done using the histological procedures. And we will talk about this in a little detail. So, let's go into animal studies. Animal studies in where there's a wide range of activities that we can do. We can study in vivo, in vitro, using microelectrodes. Microelectrodes are extremely thin electrodes which are inserted into the brain. And then we measure the electrical activity, and I'll go in the details of these a little later. Certainly, in animal studies, it is invasive. Certainly, it involves manipulation of the brain. Certainly, it involves changing the existing status of the brain, the normal status of the brain, and the normal functioning of the brain. It is, you may use evoked potentials, you may use self-stimulation, we'll come to that later. And what is basically we're trying to do is to develop animal models of the brain pathology or the brain normal functioning. And from these animal models, we move into or extrapolate into the human brain functioning. Most of the human studies are post-mortem. Of course, histological studies in animals are also post-mortem. You kill them, decap them, take their brains out, and then study the brains, how they have reacted to the earlier stimulations. But in humans, most of the studies that have been reported are post-mortem. And remember Paul Broca's brain, and I told you that he did a longitudinal study of patients who had strokes. So that was post-mortem. In Alzheimer's, patients who have Alzheimer's, their brain is taken out and studied post-mortem to see the changes that have taken place in the normal brain. Post-lesion, post-damage, we don't damage the brains. We cannot. Ethically not possible. But we do, when these patients die, take their brains and study them for the benefit of mankind. And there are many famous cases. The case of Phineas Gage, I remember I told you, where this patient had a rod pass through his eye and in the frontal lobe. And he has been the subject of study. And similarly, I did talk about HM as well. The man who lives in the present only has a 5-minute memory, not longer than that. And that is also because certain parts of this brain degenerated. So we have these post-mortem studies that are going on. Neuroanatomical studies are mainly, if you want to study the structure and the functions of the brain and the neuroanatomical sites, they're mainly invasive and mainly histological, because you have to do histologies to see the structure. What is histology? I mean, I've been talking about histology. What exactly is histology? Histology is the study of brain cells after removing them from the skull case, the cranium, and cutting sections of the brain, slices, and thin enough slices which can be stained using various techniques. And I will go into the details of these a little later. And very interesting techniques, because each one of these techniques give us a different picture of the same cell and the same structures. Of course, this is invasive, where we create a lesion or introduce radioactive substances or stimulate, or in a combination of 2 or 3, and we note the behavior changes, and later on we study the brain histologically. So it's a combination of the studies and a combination of techniques that we have used. So combination means you create a lesion in a normal brain, in a normal rat brain. Lesion means to damage, degenerate. Or we introduce radioactive substances within the brain, or use stimulation, electrical stimulation, pulsate the brain with electrical charges so that changes are behaviorally seen. And then we change this behavior, we note it, and then later on we study it histologically. Now, histology. Why is histology needed? After an experiment, why do we need histological procedures? Why do we need to study what changes have taken place? Of course, you are logical and you understand. To verify that what we have said we have done, there is evidence within the cell that this is what we have done. So, the histology is needed for the verification of location of the lesion, of the location of the stimulation sites where we said that we have electrically charged the neurons by using electrodes, and by doing so, how do we address and verify, provide evidence that we have been in the same location that we were talking about. So, it verifies the cells, the fibers, the connections after the experiments are over. They are also used for identifying locations, structures, and the functional organization of the central nervous system. And there are various techniques within this group, and I'll give you some of them. But the procedure is, the first step in this, after the experiment is over and the rat is ready for a decapitation, before we do that, we call it perfusion. Perfusion is done or carried out. Perfusions. Perfusions and perfusion of the circulatory system. This is, if you have a weak heart, strengthen yourself. I've been doing it all my life. You take a rat, open the rib cage, take out the heart, and put a needle with saline into the ventricle. And the heart of this anesthetized animal pumps away. And after the saline is 0.5% salt. You do know that your blood is saline. Every time you cut your finger, you automatically put your finger in your mouth and you taste the salt. That is 0.5%, 9% saline. So, putting saline through the heart, through the circulatory system, cleaning the circulatory system, then adding through the syringe the formalin. Now, what is formalin? Formalin is a fixative. It fixes the heart, the fibers, the systems, the tissues in exactly the form that we want them at that point in time. So, the second step is fixating the systems, the organism systems. And we use formalin, and this is called actually formaldehyde. Formaldehyde to prevent the degeneration of tissue, and it also fixes the protein bonds. So, first step is remove all the blood. The second step is add a fixative such as formalin. And the third step is therefore then to harden the tissue. You take out the brain and harden this little rat brain that you have. The rat brain is not bigger than this, the two fingers that I have, between the thumb and the finger. So, take the rat brain and harden it. And how do we harden it? We put it on a microtome. A microtome is an instrument. If we place the brain on it, it is called the freezing microtome. The freezing microtome will immediately freeze the brain into somewhat solid structure. And then this solid structure we use the cutting knife to cut thin slices. After we cut the thin slices, we put these thin slices onto glass slides, and then we stain these glass slides with the kind of stains we are interested in. Staining the cells, there are many stains. With each one of these stains means color, dye. So we dye the brain slices with certain colors using certain techniques to find out more about the structure. And some of the names that I'm going to give you are what you've heard in the first 2 or 3 lectures when we're talking about the history and development of behavior neuroscience. So staining the cells is a process in which many stains are our choice. And what we are going to be doing is that you're taking a typical characteristic which will bring out the different structures and different parts of the cell. Nissl blue. Nissl blue is a technique which, of course, blue as in the background. It stains the cells blue. Now, cell soma stained blue. So, which means you can identify the cell bodies. These little dots all over the slide are these cell bodies, and I'll show you some pictures there too. Then there is a Weil stain. Weil stain is a stain which, now cell is stained here, this is blue, but there are projections of the cell, the dendrites, the axon, which are coated by a fatty sheath called the myelin. And what the Weil stain does, it does not stain the cell, but it stains these projections wherever they are, wherever they are going. So very interesting, you can use a combination of the Nissl and the Weil to identify the cell body and then identify the cell projections. Then there is a Golgi-Cox silver staining, which is where you're using, remember Golgi? Yes, he is the one who got a Nobel Prize for giving us this technique. Heavy metals such as silver is absorbed by the tissue, we know that. And then the Golgi technique gives us those cells. So, one whole cell, Golgi will give you one whole cell which is stained fully and completely. But for some strange reason, the Golgi-Cox silver does not stain all 100% of the cells in which we have injected this substance. Only 2 to 3% neurons are stained, but they're stained completely. So, dendrites, soma, axon, right up to the mini branches are stained. So, you can see these little cells stand out as if in the background. It's like a beautiful sight to see cells with their branches. Then there is another technique which is called dry formaldehyde gas histofluorescence exposure. Histofluorescence is a relatively new technique in which we expose the cells with dry formaldehyde gas and the cells fluoresce. You know what fluoresce is? They emit fluorescence. These fluorescence tubes in your home, look up and you can see these fluorescence tubes. They're emitting fluorescence. So similarly, very interestingly, cells also emit fluorescence and you can pick up their chemical compositions are different, so their fluoresce is different. Some will fluoresce yellow, some will fluoresce green, some will fluoresce blue. So you can see the cell groups and you can identify them. And this is a technique which was brought in the 60s, in the late 60s. It's one of the very useful techniques. Then there is another group of histological procedures, the neuroanatomical tracers. Tracers means tracing the pathway. Of course, understandable. And there are various tracing methods which we follow up on projections. Where is this going? Where are these going? Where are these going? There are several types of tracers which are used. There are one in which we can inject into the cell body, and from the cell body the tracer moves outward to other cells. So it is injected into the cell body and the tracer then moves outside, and this is called the anterograde tracer. The anterograde tracer which traces from the cell towards the outside of the cell. And then of course there is a retrograde. A retrograde means, retrograde you inject here somewhere, and when you inject here, then it moves backwards to the cell. Retro. Retro means going backwards. So it goes backwards towards the cell soma. Why? Because it will show the cell and its interconnections. There are many techniques that we have used. Autoradiography is one. Horseradish peroxidase. Have you heard the name horseradish before? No, you don't think so? You don't think so? जब आप मूली खाएं तो याद करें उसको हॉर्स रेडिश कहते हैं. उसके जो एंजाइम्स हैं वो इतने पावरफुल एंजाइम्स हैं that those enzymes are used in tracing the tissue of the brain. अब जितनी दफा आप मूली खाओगे याद आएगा हॉर्स रेडिश पेरोक्सीडेज यूज होता है ब्रेन के ट्रेसर्स के लिए. इसी तरह से ऑटो रेडियोग्राफी. ऑटोमेटिक, सेल्फ. रेडियोग्राफी का मतलब रेडियो लेबलिंग. और ग्राफी का क्या मतलब हुआ? ग्राफिक is writing, graphic picture, writing, self. So self, radio labeled writing or picturization. Autoradiography is so simple to understand. It's an interesting procedure because it traces different pathways for you. Similarly, horseradish peroxidase also gives you a very nice tracing mechanism. Then there are the radiolabeling. What are the radiolabeling techniques that we use? The radiolabeling techniques that we use, of course, you in this modern age know what radioactivity is, right? Where you have added radioactivity into a system. No. We don't do too much of radioactivity into the brain. Very small amounts of radioactive, only to tag and only to show up during the... So it's not dangerous, but at the same time a useful study. Carbon-14, which enters the cell systems, or 2-DG glucose. Now, glucose is used by every single cell, and it is extremely important to provide energy to the cells. Therefore, wherever cells are going to be active, glucose is going to be used. So what do? As wise as the neuroscientists are, they radiolabel the glucose. So wherever brain activity is higher, I'm talking now and thinking, so my talking area will show up if I'm injected with 2-DG glucose. Or my thinking area will brighten up and show up with this radiolabeling. Very interesting. And these are used they enter the cell by mechanism of its own metabolism, and it stays there. Because it's radiolabeled, the radioactivity does not go. Eventually it does get dissolved, but excreted, but right then and there it is sitting in the system. So, it emits radioactivity when exposed to a photographic plate. So, they take this brain slide which has been exposed to radioactivity and you place it on top of a photographic plate, and the photographic plate will show up just like a black and white negative. This activity will show up, and this is a post-mortem study, but you will see that 2-DG glucose is also used in various scan studies, the MRIs and the PET scans, etc. The amino acids or amines for, they are used for amine, the neuroamines and the neurotransmitters. They are also radiolabeled. So wherever dopamine is being used, the radiolabeled amine for dopamine will enter there. For serotonin, it will go into another place. For norepinephrine, it will go. So you have the capacity to tease out the various neurochemicals and their bunches and their projections and their groups by using radiolabeling activity. So neural tissue, when exposed to a label ligand, then these ligands bind to a target and then they show up in the radioactivity. Very interesting. Simple, well thought out, clean, and gives us clear evidence. The HRP or the horseradish peroxidase enzyme, which is a retrograde exoplasmic. Retrograde means move back into the cell. But this is exoplasmic. So it moves from the axon back into the cell, not from the dendrites. Exoplasmic moves from the plasma of the cell through the axon into the cell. Aha. So exoplasmic retrograde transmission, HRP or horseradish peroxidase. मूली याद रखें. And so that enters the cells and then moves on to the dendrites. So this has entered and stained, and then it will show up in the subsequent fluorescence. What does it do, this HRP? It is capable of breaking down certain peroxide molecules into soluble salts. These are then taken up by the terminal butons. अभी लिख लो terminal butons. बाद में मैं 2 lessons के बाद 2 lectures के बाद फिर समझ आएगी. Terminal butons ये हैं. Every time a cell talks to another cell, it talks through the terminals. So terminal butons, the synapses हैं, उनको भी वो stain करता है. The single cells then stain and they glow and fluoresce under certain wavelengths. So इसको कहते हैं histofluorescence. Very interesting. And this technique was developed by Falk and Hillarp in the 1960s, Swedish researchers. Then there is another technique, which is Immunoassay Chemistry. As the name implies, it is introducing an antigen to create antibodies, monoclonal antibody, single clonal antibodies, and they can therefore, when you're talking about monoclonal antibody, you know the antigen that you're looking for and injecting and the kind of antibodies that you are trying to create. So you must know your cell, you must know your chemistry very well, which means the chemistry of the brain and the location that you're looking for. You can use specific proteins for specific neurotransmitters, for specific regions of the brain. And what we are hoping and what we see is there is an accumulation of radioactive material or dyes which show up under the microscope as when we look at the location of the neuroprotein. So, basically, simple process, you know you are creating antibodies and you're looking for these antibodies. जिस तरह आपको बीमारी के लिए टीका लगता है ना, टाइफाइड का, तो टाइफाइड के जरासीम अंदर खुद पैदा होते हैं, वो अपनी रेजिस्टेंस पैदा करते हैं और वो इम्यून सिस्टम को अराउज करते हैं. तो ये जब बनते हैं सेल्स, ये एक जगह जाके जमा होते हैं, क्लम्प होते हैं. So we are looking for labeled antibodies for cited, target cited ligands. Ligands are those locations, those chemicals जो वहां जाके attach हो जाएंगे. So immunocytochemistry is a very specific histochemical procedure. And so I've given you histochemical procedures, and there are many interesting histochemical procedures, and each one of them has their benefits and has what it stands out for. One will stay in the cell, one will stay in the cell bodies, one will go anterograde, one will go retrograde, one will give you a bunch of neurons which fluoresce in a different way, so you know exactly where the projections are. So this is one group of techniques that we can use. Now, if you're looking for neuroanatomical techniques traditionally have been very effective, and they have been used to identify sites, to identify the brain functions. So essentially, this is invasive. And this means that you change what is normally happening within the brain. You can do it many ways. You can activate the brain by using electrical stimulation. Very interesting idea. Electrical stimulation. ये आपने साइंस फिक्शन की फिल्में देखी है ना, जहां पर वो इलेक्ट्रोड लगे हुए हैं और वो फ्रैंकस्टाइन चलता जा रहा है. Actually, this is what happens in the laboratory. We don't create Frankensteins, but we do put electrodes in the rat brains, and I'll show you some pictures where you will see this happening. And I've done it in a lifetime, many rats, many locations. So electrical stimulation, where you insert electrodes in brain. So you go in the brain and insert an electrode, and then you charge this electrode, और पावर यहाँ जब जाता है electrical charge, तो the brain is activated, this particular area is activated, and the rat, whether you want the rat to move, to sit, to aggress, to eat, the behavior follows. Very controlled. Electrodes are implanted. You know, implanted means put inside the brain. And they're stereotaxically implanted in the brain. And they're implanted using very specific techniques. We use the Stereotaxic Atlas, and we use the Stereotaxic apparatus, and we use the electrode fixating apparatus, and of course we use the electrodes, and of course we use a mechanism by which electrical charge can be passed into the brain. Science पूरी. आपका क्या ख्याल है जब ब्रेन सर्जरी होती है हॉस्पिटल्स में, तो क्या होता है? Exactly the same procedure. For human brain surgery, stereotaxic atlases are also used. Atlas कैसी होती है? Atlas वो है जिसमें आपको लोकेशन, जिस तरह ज्योग्राफी की, आपने देखना है कि आप मेडागास्कर कहाँ पे, तो आप कैसे देखते हैं? एटलस को देखते हैं ना. एटलस बताता है लॉन्गिट्यूड इतना, लैटिट्यूड इतना, वहां जाके आप पहुंचेंगे तो मेडागास्कर होगा. मेडागास्कर का नाम नहीं सुना ना, लेकिन आप एटलस में ढूंढेंगे तो आपको पता चल जाएगा. इन द सेम वे, you want to get to, let's say, hypothalamus. You can't see hypothalamus here, but in the brain atlas, it is stated, if you go so many millimeters to the left, so many millimeters down, and so many millimeters within. So there are 3 dimensions, and I'll give you those 3 dimensions. Interestingly, this apparatus very scientifically projects exactly what you see on the atlas. So atlas में देखते हैं कि हमने किधर जाना है, और फिर हम अपने इंस्ट्रूमेंट के साथ उस लोकेशन में पहुंचते हैं और इलेक्ट्रोड वहां इंसर्ट करते हैं. इलेक्ट्रोड होता क्या है? इलेक्ट्रोड एक फाइन नीडल है की सारी इंसुलेटेड है। इंसुलेशन क्या होती है बता? इंसुलेशन वो आप बिजली की तारें अपने इर्द गिर्द देखें, दैट इज इंसुलेशन। हर तार के ऊपर रबर लिपटा हुआ, राइट, इंसुलेट हुई भी, लेकिन जहां से कनेक्शन उसका बनना है वहां पर वो अनइंसुलेटेड है, वो प्लग के अंदर अनइंसुलेटेड है। इसी तरह से ये इलेक्ट्रोड जो है, ये पूरे की पूरी सुई जो है, नीडल इज insulated except for this very small 0.5 millimeter tip which is then inserted। जब ये टिप अनइंसुलेटेड है, जो इलेक्ट्रिकल चार्ज यहाँ से आएगा यहाँ तो नहीं लगेगा क्योंकि ये इंसुलेटेड है, यहाँ पर जाके वो चार्ज जो है ना पल्सेट करेगा। जब वो पल्सेट करेगा तो एक्टिविटी उस एरिया में जाएगी। सो 0.5 millimeter uninsulated tip allows the current to pass through the machine, through the wire, through the electrode into the brain. What is the stereotaxic instrument? Stereotaxic instrument is an instrument which allows placing of electrodes and other surgical procedures using precise positioning. Now, what is this precise positioning? There are three dimensions of this precise positioning. One, the anterior posterior. अब ये भी थोड़ी सी टर्म समझ लें। Anterior means आगे की तरफ, posterior means पीछे की तरफ। So इस तरफ आना है या इस तरफ जाना, towards the front or towards the back. So anterior and posterior. The second is dorsal or ventral, ऊपर की तरफ या अंदर की तरफ, गहराई की तरफ। दो डायमेंशन बड़ी आसानी से समझ आ गई। तीसरा डायमेंशन है कानों की तरफ या मिड लाइन नाक की तरफ। सो मिड लाइन को कहेंगे मीडियल और कानों की तरफ होगा लैटरल, टुवर्ड्स द लैटरल साइड्स। ये तीन डायमेंशन इस्तेमाल होती हैं तो ब्रेन सर्जरी हो जाती है। चाहे ब्रेन सर्जरी ह्यूमन में हो, चाहे ब्रेन सर्जरी रैट की हो, चाहे डॉग की हो, चाहे हॉर्स की हो, चाहे एप की हो, ये तीन आपकी डायमेंशन इस्तेमाल होती हैं। सो वन इज एंटीरियर पोस्टीरियर, वन इज डोर्सल वेंट्रल which is on the top or inside, and then there is medial lateral which is this way. There is in the stereotaxic instrument a head holder. You can't move the head because you've made exact measurements. The head is stuck. और चूहे का जो ईयर बार और जो दांतों को है उसको फिक्स करते हैं ताकि किसी तरफ से वो मूव ना करे हेड, इट्स फिक्स्ड। And this system only allows the electrode to be moved or electrode holder to be moved and follow exact measurements. Stereotaxic atlas is like the geographical atlas. Dimensions are given to arrive at a particular location. And there are sutures in the skull. Sutures means there are इस तरह जोड़ बने हुए हैं स्कल के अंदर, खोपड़ी के ऊपर जो आपकी हड्डी है खोपड़ी की, उसके ऊपर निशान सूचर्स बने हुए हैं। और रैट्स में हम इसको यूज करते हैं। इसको दो हमारे पास सेक्शंस हैं, एक है एंटीरियर के लिए ब्रैग्मा, BREGMA लैटिन में है, और दूसरा है पोस्टीरियर की तरफ लैम्डा, LAMBDA। ये हर एटलस में आपको ब्रैग्मा और लैम्डा नजर आएगा। आप सिर्फ देख लीजिए, आपको इससे ज्यादा जरूरत नहीं याद रखने की। And there are specific atlases for specific species. So अगर मैंने रैट सर्जरी करनी है तो रैट की ब्रेन एटलस देखूंगी। स्टीरोटैक्सिक एटलस अगर मैंने मंकी ब्रेन पे सर्जरी करनी है तो मंकी स्टीरोटैक्सिक एटलस देखूंगी। अगर मैंने ह्यूमंस पे करनी है तो ह्यूमन ब्रेन स्टीरोटैक्सिक एटलस देखने की जरूरत होगी। So we use these stereotaxic instrument, stereotaxic जैसे उसकी डिस्क्रिप्शन है, then you use the stereotaxic atlas, and of course using this you can do many variety of surgeries within the animal research। और इसमें हम स्टिमुलेट कर सकते हैं, इलेक्ट्रिकल स्टिमुलेशन कर सकते हैं, केमिकल स्टिमुलेशन कर सकते हैं, पुश पुल कैनुला डाल सकते हैं, interesting, and there are no real ethical questions here. Of course, you have ethical questions, but you make sure that you follow all humane procedures for doing surgery in animals. In the next class, we'll go into the details of the electrical stimulation, the chemical stimulation, and also using the push-pull cannula. And this is what we are doing in the invasive study of neuroanatomical sites of the brain. Interesting, because this has given us an extremely valuable insight into the functioning of the brain, and we have seen that rats, cats, dogs, humans all behave alike if stimulated in a similar area. Details in the next class, but do read your text and do catch up on your National Geographic as well. And if you get website of neurosciences, extremely valuable. Next time I'll bring in several sites for you so that you can look up those websites and check out more about neurosciences if you're interested.